Imagine a vast sweeping landscape, maybe the plains of Africa, you know, dotted with cattle. Now, zoom in on just one cow. Its ribs showing, head hanging low, just looking really weak. What if I told you that hidden there, often microscopic, there's a threat quietly undermining global food security and, well, the livelihoods of millions?
It's a huge challenge, one many people don't even know exists.
Yeah.
But its impact is absolutely staggering.
Staggering, yeah, and like you said, largely unseen. That's exactly why today we're diving into bovine trypanosomiasis. Most people know it as Nagana. And this isn't just some, obscure animal disease. It's a debilitating parasitic condition in cattle that has really far-reaching, complex, and frankly surprising global implications.
Right. Our mission today is to really unpack this. It's a layered disease. We'll trace it from the tiny culprits, the protozoa,
All the way up.
all the way up to its huge economic footprint. We want to reveal some crucial insights, things often overlooked. Ultimately, we want you, the listener, to understand why this isn't just a vet problem. It's a critical challenge impacting millions of people's lives.
Okay, so to really get a handle on this, we need to start small. The tiny architects, as you called them. So what is Nagana, fundamentally?
Fundamentally, Nagana is a parasitic disease. It's caused by these single-celled organisms, protozoa, from the genus Trypanosoma. Now, while it's infamous, for being a massive constraint on livestock in Africa.
Yeah, that's where I mostly hear about it.
Right, but it also causes really severe economic losses in parts of Asia and South America, too. It's definitely not just an African issue.
Okay, Trypanosoma. You said species, plural. Are they all equally bad, or are there main players we should focus on for cattle?
That's a great question. You don't need to memorize all the names now. But understanding their different types is really key. It's kind of like different flu strains. Some hit harder. For cattle, the main ones causing severe disease are Trypanosoma congolens. That one's generally the most pathogenic, the nastiest.
Okay.
And Trypanosoma vivax. This one's notable because it's pretty versatile in how it spreads.
Yeah.
Then there's also Trypanosoma brucei. Less severe in cattle usually, but a big problem for horses.
Right.
But yeah, in African cattle, it's really T. congolens
Mm-hmm.
and T. vivax that cause the biggest economic headaches.
But hang on, I think I've heard of others, ones that spread beyond that tsetse fly belt everyone talks about.
You're asking a really vital question there.
Yeah.
That brings us to Trypanosoma evansi. That's the agent Asura.
Yeah.
This one's fascinating because it has the widest host range and the widest global distribution. It can thrive even in drier zones, way outside the tsetse areas. A real globetrotter.
Wow. Okay.
And then there's T. thaleri. You find it pretty much worldwide. But the key thing is it's considered non-pathogenic. So not really a cause for disease concern.
So it sounds like trypanosomiasis in cattle isn't just one single thing. It's more like a family of diseases.
Exactly. That's a vital point people often miss. Nagana isn't monolithic.
And why does that distinction matter so much? Does it change how we fight it?
Oh, absolutely critical. Because what works for one type, say controlling the tsetse fly for T. decongolens, might be totally irrelevant for T. vatsi spreading somewhere else. You need tailored strategies. A blanket approach it just won't cut it. Recognizing this difference is paramount for any effective management.
Right. Okay.
Yeah.
Which logically brings us to how it actually spreads. Most people, when they think Nagana, they think tsetse fly.
Yep, that's the classic image.
And they're not wrong, but clearly that's only part of the story, isn't it?
It is. Tsetse flies, the Glossina species, are the primary biological vectors. That means the parasite actually develops inside the fly. They transmit it through their saliva when they bite an animal to feed. And yeah, they're largely confined to that tsetse belt in sub-Saharan Africa. Their geography dictates where those specific forms are endemic.
Interesting.
And just a detail, T. vivax actually has the shortest development cycle inside the tits. Makes it pretty efficient.
So that biological link, that dependence, it means if you can control or even wipe out tsetse flies in an area, you can potentially eliminate the specific trypanosomes there.
Potentially, yes. In confined areas, tsetse eradication can eliminate those forms. It's a powerful direct approach, and it has worked in some places.
I sense a but coming.
But, well, that's definitely not the whole story, not by a long shot. And here's where it gets really interesting and honestly a bit more alarming. We need to talk about the hidden global threat. Mechanical transmission.
Mechanical, what does that mean?
It means the parasite is just physically carried on the mouth parts of biting insects.
Yeah.
It doesn't develop inside them.
So not just tsetse flies?
Nope. This is how parasites like T. vivax, and especially T. evansi, spread far beyond the tsetse belt. We're talking about other biting insects, tabanids, those big horse flies.
Ugh, yeah.
Stable flies, Stomoxys species, maybe even mosquitoes sometimes.
Wait, so those common biting flies can spread it too. How does that work exactly? Is it as effective as the tsetse?
Well, it happens when an insect starts feeding on an infected animal, gets interrupted, maybe swatted away, and then quickly moves to bite another nearby animal.
Yeah.
It transfers the infected blood directly on its mouth parts. The efficiency is highest when that time gap is really short, like less than 30 minutes, before the blood dries or clots.
Wow.
And this mechanical transmission is precisely how T. vivax and T. evansi have managed to establish themselves globally. We see them in places totally free of tsetse flies South and Central America, the Caribbean.
Seriously.
Seriously. And increasingly, we're seeing reports from places like Australia, Spain, even France.
That completely changes the game. This is genuinely a global problem then, not just centered on Africa.
Absolutely.
And I bet controlling those other flies, the mechanical vectors, must be a nightmare compared to the tsetse. They're everywhere.
It is an immense challenge. They're incredibly diverse. They're highly mobile. They reproduce like crazy. Eradicating them is basically impossible over large areas. So for these mechanically transmitted forms, the focus shifts. It's less about eradication and more about continuous, localized management trying to keep their numbers down.
Okay, so tsetse, other biting flies. Are there even more ways this thing spreads? Any surprising ones? Maybe things we do?
You've hit on a really critical point. There's what we call iatrogenic spread. That means human-influenced. Through non-sterile equipment during routine farm work. Think about mass vaccinations, dehorning, castration, ear tagging, even something like rectal palpation for pregnancy checks. If you use the same needle or the same instrument on multiple animals without sterilizing it properly in between.
You can just pass the parasite along.
Exactly. You inadvertently become the vector. It underscores how vital basic biosecurity is. This is preventable.
That's quite sobering. Something as simple as using a fresh needle.
It makes a huge difference. Then there are other routes, too. Direct horizontal contact between animals, though how significant that is, isn't totally clear.
Yeah.
There's vertical transmission from mother to fetus, transplacental, which can cause abortions or infected calves,
Oh, wow.
and per-oral contamination, mainly relevant for carnivores eating infected meat. And just a quirky one in Latin America, vampire bats can play a role.
Vampire bats. Seriously.
Yep, they can ingest infected blood, become carriers, and then transmit it when they bite other animals, both reservoir and vector.
Okay, mine's slightly blown on the vampire bats. So we know what it is, how it spreads.
Yep.
What is not going to actually do to the cow? What are we looking at clinically?
Well, the incubation period varies. It can be as short as 4 days or up to 8 weeks. For the tsetse transmitted types, it's usually quicker, maybe 1 to 4 weeks. Early signs, often an intermittent fever, up and down. Severe anemia is a big one. You'd see pale gums, pale membranes around the eyes.
Okay.
And just general lethargy, lack of energy, and progressive weight loss. The animal just starts wasting away.
And I read something interesting, a little detail for you listening. Sometimes an initial swelling, called a chancre, can develop right where the fly bit. But here's the catcher. Farmers often don't even notice it. It's small, maybe hidden by hair. So spotting it early in the field, really tough.
That delayed recognition is a massive problem, yeah.
Yeah.
Then the disease can progress in a couple of main ways, acute or chronic. The acute form hits fast and hard. Really severe symptoms pronounced anemia, maybe generalized swelling or edema, lots of tearing from the eyes. Often leads to death pretty quickly, especially in animals that haven't been exposed before, like imported breeds.
And the chronic form, is that the one that just grinds them down slowly? The sort of silent drain?
That's exactly it. The chronic form is actually much more common in ruminants. It's a long, drawn-out process. You see this gradual but severe wasting and a marked drop in productivity. This is where that huge hidden economic burden comes in.
Right. It's not just the animals dying.
No, it's far beyond that. It's the insidious losses.
Yeah.
Less milk, neurological problems sometimes, dependent edema, swelling in the lower legs, and brisket heart problems, even cloudiness in the eyes, corneal opacity. The impact just ripples outwards.
Wow. Are there any other, maybe more extreme ways it shows up? Any unusual clinical pictures?
Oh, absolutely. And here's where it gets particularly nasty with certain strains. There's something called acute hemorrhagic syndrome.
Hemorrhagic. That sounds bad.
It is. It's a distinct, rapidly fatal condition. We see it sporadically, mainly with specific T. vivax strains, often in East Africa. Animals get enlarged lymph nodes, incredibly severe anemia, and widespread bleeding, internal hemorrhages, bleeding from the nose, in the gut. In some outbreaks, they even see bleeding from the ears. It's thought to be caused by the immune system going haywire and destroying red blood cells. It really highlights how variable even a single Trypanosoma species can be. Not all TB-vax infections look the same.
That's incredible variability. What about age? Does it affect young animals differently? Are they tougher?
Interestingly, yeah, often they are. A bit like we see with other tick-borne diseases like anaplasmosis or babesiosis. Younger animals, calves, they might show greater natural resistance to the clinical disease. They can get infected, sure, but they might not get as obviously sick. They can become carriers without showing those severe signs.
Okay, so we have this complex enemy. It can be acute, it can be chronic, it spreads in all these different ways. How on earth do you actually diagnose it reliably? It sounds tricky.
It is tricky. Accurate diagnosis is crucial, obviously, and the test you choose depends on things like cost, what equipment and expertise are available, and are you looking at one sick cow or trying to screen a whole herd.
Right. So walk us through the diagnostic toolkit. What are the options?
Well, the simplest, often the first step, is basic microscopy. You can take a drop of blood, put it on a slide, a wet blood film, and look for moving parasites. It's quick, immediate.
But not very sensitive.
Exactly. Low sensitivity, especially if the animal doesn't have many parasites, like in chronic cases. Then you have stained blood films. Giemsa stain thin and thick smears. These let you see the parasite's morphology better, maybe even identify the subgenus. But it takes time to prepare and read them, and sensitivity is still an issue.
So for those tougher cases, low parasite numbers, how do you boost the chances of finding them?
Then you move to concentration techniques. These basically concentrate the parasites from a larger blood sample. Methods like the hematocrit centrifuge technique, the HCT, often called the Wu test, or the Buffy-Toad technique, BCT. You spin down the blood, and the parasites concentrate in a specific layer.
Oh.
The HCT is neat because you also get the packed cell volume, the PCV, right away, which tells you how anemic the animal is. There are even more sensitive ones like MAECT, but they tend to be more cumbersome and expensive, less practical for everyday field use.
Okay, so microscopy, concentration. What about the high-tech stuff? The gold standard for precision.
Right. That brings us to molecular methods, mainly PCR polymerase chain reaction.
The DNA test.
Essentially, yes. PCR is incredibly specific and sensitive. It can detect parasite DNA even when there are very, very few parasites present, crucial for finding those silent carrier animals. It's super important for understanding the epidemiology, like which strains are where, and for monitoring the nightmare of drug resistance.
And sample collection is easier now?
Yeah, that's improved. You can often just collect blood spots on special filter paper, easy to store, easy to transport to a lab. The downsides: you need specialized labs, trained people, and there's always a risk of contamination leading to false positives if you're not careful.
Okay, so PCR finds the parasite itself. What about finding evidence that the animal's immune system has seen the parasite, even if it's not sick right now?
For that, we use serological tests, things like IFAD immunofluorescence antibody test or ELISA enzyme-linked immunosorbent assay. These look for antibodies the animal produced in response to infection. They're really useful for finding animals that have been exposed or might be carriers, because you don't need the live parasite to be circulating. Great for large-scale surveys.
But there's a catch, right?
There is. Antibodies can stick around for a long time, even after the infection is cleared. So a positive antibody test tells you the animal was exposed at some point, but it doesn't definitively confirm an active current infection.
So putting it all together, it sounds like you rarely rely on just one test for a definite diagnosis.
Pretty much. Especially because the clinical signs can be so vague, so nonspecific. Even post-mortem findings aren't always clear-cut. You often need to combine the clinical picture with maybe microscopy, maybe a concentration technique, maybe PCR or serology to really nail down what's going on. It's often a multi-pronged diagnostic approach.
Okay, so we've identified the enemy, we understand its attacks, we know how to find it. Now the really big question, how do we actually fight back? What are the treatment and control options against something this, well, insidious?
Right, the fight back. Well, starting with treatment, the arsenal of drugs, the chemotherapeutics, it's actually quite limited, worryingly so. The main players are dimetazine acetate, that's mostly for treatment, and isotimidium chloride. Isotimidium is interesting because it can be used for treatment and has some preventative or prophylactic activity.
Okay, so two main drugs. Are there others?
There are a few others like homodium salts, but dimetazine and isotimidium are really the workhorses in the field.
This immediately raises a question for me. If the arsenal is so limited, what are the challenges? It surely can't be as simple as just giving a shot.
Oh, definitely not.
Yeah.
There are significant challenges. Firstly, most of these drugs have a narrow therapeutic index,
Meaning?
meaning The gap between a dose that works and a dose that's toxic is quite small. You have to be careful with dosing.
Right, that's risky.
Very. And the other massive growing concern is drug resistance. Especially to diminazine, we're seeing more and more resistance reported, leading to treatment failures. That's a huge problem.
So the drugs might just stop working.
In some cases, yes, or work much less effectively. It's also worth noting, and this is important for farmers, well-nourished animals generally respond better to treatment. Their overall health status really matters.
Okay, so treatment has its issues. What about prevention using drugs? That prophylactic effect you mentioned with isomimidium, chemoprophylaxis.
Yeah, that's the idea using drugs to provide protection over a period of time. Isomimidium chloride is the main drug used for this. But again, it's tricky. You need really precise administration intervals, give it too early or too late, and you lose protection or risk resistance. And combining drugs can be dangerous. For example, if you give diminazine for treatment shortly after an animal has had several preventative doses of isomimidium, you can actually cause severe liver damage, even death.
Wow, okay, so you have to know the history.
You absolutely do. And how long the protection lasts also depends heavily on the challenge, basically. How many infected flies are biting the animal? High challenge means the drug gets used up faster.
Any new drug tech on the horizon? Slow release, maybe?
People have experimented with slow release devices, yes. But so far, they've often come with concerns about potential long-term side effects, like being mutagenic. So not a widespread solution yet.
Okay, drugs are crucial but complicated. What about going after the vectors? Especially that tsetse fly. It seems like a logical target if it's biologically linked.
Vector control is absolutely a cornerstone, yes. But the approaches really differ depending on which vector you're talking about. For tsetse flies, control can be highly effective, precisely because they're often confined to specific geographical areas or belts.
So what kind of methods work for tsetse?
A range of things. You can use insecticide-treated cattle, the flies land, pick up the insecticide, and die. There are tsetse traps and
I've
targets.
seen pictures, those blue cloth things.
Exactly. Often blue cloth, sometimes with black panels. They're attractive to tsetse, often combined with olfactory attractant smells like acetone or even aged cow urine to lure them in. The traps can be coated with insecticide or designed to capture them.
Clever.
Then there's aerial or ground spraying of insecticides in specific habitats, and sometimes modifying the habitat itself, like clearing dense bush where flies breed.
And what about that sterile insect technique, SIT? That sounds pretty advanced.
It is quite specific, and it's environmentally friendly, non-polluting. The idea is you mass rear huge numbers of male tsetse flies in a lab. You then sterilize them, usually with gamma irradiation, and release them into the wild in overwhelming numbers.
And the sterile males?
They mate with the wild females. But because tsetse females typically only mate once in their lives, these matings produce no offspring. Over time, this crashes the wild population.
Ingenious, but slow and hard to do.
It has a delayed effect compared to spraying insecticides, yes. And mass-rearing tsetse flies is challenging. They reproduce very slowly, only one larva at a time. It's not like mass-rearing fruit flies.
Okay, so lots of tools for tsetse, but back to those other flies, the mechanical vectors, the horse flies, stable flies, spreading T. vivax and T. avanci everywhere. Can you even control them effectively?
It's considerably harder. Much, much harder. Their sheer diversity, their ability to fly long distances, their rapid reproduction, ecological control over large areas is often just not feasible or sustainable.
So what do you do?
For those, the focus really has to be on continuous localized management. Things like fly traps around barns, maybe targeted insecticide use in specific areas, keeping manure cleaned up where stable flies breed. You're aiming to reduce their numbers locally, not wipe them out across landscapes. It's a different mindset.
Right. Beyond drugs and fly control, what about managing the cattle themselves? Things like breeds or that biosecurity point you made.
Absolutely vital parts of the puzzle. Breed selection is a huge one. There are breeds of cattle, particularly West African taurine breeds like the Ndama, that have this remarkable natural genetic resistance or tolerance to trypanosomiasis. They can get infected, but they control the parasites better and don't get nearly as sick.
So using resistant breeds is a strategy.
A very powerful one, yes. Now, the resistance isn't absolute. It can break down the really heavy fly challenge or if the animals are stressed or poorly fed, but it's a major genetic tool.
And biosecurity. Remind us why that's key.
Back to that iatrogenic spread. Rigorous sterilization of absolutely all veterinary instruments, surgical tools, castration knives, everything. And crucially, using a new sterile needle for every single injection or blood draw. It sounds basic, but preventing that human-driven transmission is one of the most impactful, controllable factors we have.
Makes sense. Anything else on the management side?
Yeah, things like controlling animal movement, especially moving animals from low-risk to high-risk areas without protection, and just ensuring good nutrition. Well-fed animals are more resilient to infection and respond better if they do need treatment.
Okay, so we have drugs, vector control, management practices, quite a few tools. But there's still this huge elephant in the room, isn't there? The vaccine gap.
Unfortunately, yes.
Yeah.
That's the massive gap. Despite the absolutely profound global impact of bovine trypanosomiasis, decades of research, there are still no commercially available vaccines for cattle.
None at all.
None. The parasite is incredibly complex. It changes its surface coat constantly to evade the immune system. It's a huge scientific challenge.
Wow.
Finding a vaccine remains a top priority, but it's proven incredibly difficult. It represents a major hole in our control toolkit and highlights just how urgently we need more R&D in this area. A successful vaccine would be revolutionary.
Absolutely. Okay, let's shift gears now and talk hard numbers. The massive price tag this disease carries. So what does all this actually mean for, you listening, for the bigger picture of global food security and economics?
Well, the economic burden is just immense, and it hits on multiple levels. If we look just at African animal trypanosomiasis AAT or Nagana in sub-Saharan Africa, the estimated annual losses are around $4.75 billion.
Billion, with a B.
With a B. And the overall impact, just thinking about the land infested by tsetse flies and the opportunities lost, is estimated around $4.5 billion annually. To give you some context, in a country like Ethiopia, authorities have stated it threatens agriculture more severely than any other single livestock disease.
That's a staggering claim. And like you said before, it's not just about animals dying, is it? There are direct losses, but also these hidden, indirect ones.
Exactly. The direct losses are kind of obvious. Animals dying, getting sick, and needing treatment, reduced fertility, the actual cost of control measures like drugs and traps. But the indirect losses, that's the hidden economic burden that often gets underestimated.
Like what?
Things like farmers having to cull animals earlier than planned, animals not gaining weight properly, huge reductions in milk production, even lower quality hides and skins. And critically, especially in Africa and parts of Asia, impaired animal traction.
The oxen pulling plows.
Precisely. If your oxen are weak from chronic nagana, they can't work effectively. Studies have shown oxen in high-risk areas can be up to 38% less efficient. Think about what that means for a smallholder farmer relying on those animals to plow their fields. It's not just an animal health problem. It directly slashes crop production. It hits food security right at the household level.
Can you give us some concrete, real-world examples, specific cases where the cost was calculated?
Sure. There was a study in the Jawi district of northwest Ethiopia. They estimated the total economic losses per household were over $1,200. The biggest chunk was from mortality, especially losing valuable oxen and milking cows. But loss of draft power accounted for nearly a quarter of the total loss, and reduced milk yield was another significant hit.
Wow, $1,200 per household. That's huge in that context.
That's devastating. Or think about a major epidemic. There was a big T5X outbreak in Brazil and Bolivia back in 1995. Estimated losses, $160 million. In India, just looking at T. avanci or Sura, the annual losses are estimated at around $671 million.
It's just relentless, these numbers.
It really is. And just for a historical comparison, looking back at U.S. data on cattle fever ticks, which cause related blood parasite diseases, the losses they cause historically would be equivalent to over $3 billion in today's money. These kinds of diseases have always been economically crippling.
And we haven't even factored in the cost of just treating the disease, the drugs themselves.
Right. Add that layer on top. Estimates suggest between 35 and 50 million doses of trypanocidal drugs are used every year just in tropical Africa. Depending on the drug and source, a dose might cost anywhere from 50 cents to maybe $3.50 or more. So you're looking at potentially up to $90 million spent just on the drugs annually.
And probably higher if you count informal markets.
Very likely higher, yes.
Yeah.
That $90 million might be conservative.
So the final economic takeaway here seems crystal clear. Investing in controlling Nagana isn't just about saving cows.
Not at all.
It's a fundamental investment in economic development, in poverty alleviation, in food security. The returns on sustainable control strategies seem massive because you're tackling something that undermines the very foundation of agriculture for millions.
That's exactly right. It's an economic development issue as much as it is an animal health issue.
Okay, so let's try and wrap this up, recap this deep dive into Nagana. We've really uncovered a incredibly complex parasitic disease, haven't we?
We certainly have. Different Trypanosoma species causing trouble.
Right, not just one thing.
Varied and sometimes really surprising ways it spreads. You've got the classic tsetse fly, but then these globally relevant mechanical vectors, and even us, humans, inadvertently spreading it through poor biosecurity, that iatrogenic route.
And we explored that whole spectrum of clinical signs, from those rapid, fatal acute forms like that horrible hemorrhagic syndrome, all the way to the chronic, insidious forms that just slowly drain productivity, the hidden burden.
And we looked at the diagnostic toolkit, the challenges, the different methods, from basic microscopy up to sensitive PCR, and why you often need a combination approach.
And then the fight back, the immense challenges in treatment and control, that very limited drug arsenal, the scary rise of drug resistance.
A huge concern.
And that massive, glaring gap, no commercial vaccine.
All of which points really to one clear conclusion. We need a comprehensive, integrated, and adaptive strategy. There's no single magic bullet.
So what does that look like?
It means differentiating control based on the specific parasite and how it's spreading locally. It means much better surveillance and diagnostics for early warning and tracking resistance.
Smarter use of the drugs we have.
Sustainable chemotherapy, exactly. Using drugs wisely to preserve their effectiveness. Plus, integrated vector control, combining methods against tsetse where relevant, and finding sustainable ways to manage mechanical vectors.
And strengthening biosecurity feels like a must.
Absolutely critical.
Okay.
Preventing that human influence spread is low-hanging fruit, relatively speaking. And underpinning everything, we desperately need intensified research and development, especially for new drugs and, crucially, for that elusive vaccine.
It really sounds like it demands that one health approach people talk about, recognizing that animal health, human livelihoods, and the environment are all completely tangled up together here.
Perfectly put. You can't solve this just by looking at the cows.
So maybe a final thought to leave our listeners with. Considering the sheer economic and social weight of a disease like Naganai, something that often operates, relatively quietly in the background compared to more dramatic outbreaks, what other hidden challenges, maybe similar slow burn problems, might be silently reshaping global economies and livelihoods right now that perhaps we should all be paying closer attention to?